Now let's take a look at some organic chemistry. We already clarified that organic compounds are those that contain carbon and hydrogen. And in addition to carbon and hydrogen, a lot of times they also contain oxygen, nitrogen, those are the popular ones. But what's different about organic chemistry or carbon chemistry is that a lot of times, well, carbon likes to find four partners, and sometimes it's not so easy to find four partners to bond with, to have these outer 8 electrons, a full shell of 8 electrons. So what carbon likes to do is just make a deal with itself. And so it forms these long chains or even ring structures that we call a carbon skeleton. So basically carbon likes to bond with itself, and you can see some here. These two carbons, they bonded with each other, and these three, they're all making a long chain. In addition, what we see in organic chemistry is that we have functional groups. So if it was just carbon and hydrogen, then we could just make endless long chains, and they would all be hydrophobic in nature because they don't have any polarity. Now, the fun comes in when we're adding some oxygen or maybe some nitrogen. Here we have hydroxyl group, the OH group that we're adding. It's also called an alcohol group, and it changes the name. If we had 4 hydrogens surrounding this carbon, it would be called methane. If we add the hydroxyl group, it's called methanol. Or here, this is a fun one, if we have just hydrogen surrounding the carbon skeleton, and it's called ethane, and it's a gas. If we add in this hydroxyl group, then it's ethanol. And this stuff is actually pretty good. This is drinking alcohol. And then here we have isopropanol, that's usually used as a disinfectant. So here, again, this OL ending comes from this OH group that was added here to the carbon skeleton. The hydroxyl group is not the only functional group that's important. So here the OH group is also called the alcohol group or hydroxyl group. Then we have the aldehyde group, that's C double bond O with an hydrogen right here. That's the aldehyde group. Then the keto group, that's the C double bond O. The methyl group is a CH3, that's a methyl group. And then we have, very important here, the amino group. This is very important for you guys. The amino group is an NH2 that's being added. And that's important because the amino group is seen in all of the amino acids that are making up all the proteins. So here, continuation. Here would be an ester group. This one, we won't be talking too much about it, but if you're making if you're combining fatty acid to make dietary fats, then you need this ester group. An ether group is a COC bond right here. Sulfhydryl, we see that in some amino acids, you have a sulfhydryl group. Carboxyl group, this one is important. So this one I would say is, oops, you should remember that. That's the organic acid group, so C double bond O and then OH, and here is a phosphate group. Okay, so let's take a look at this. So we just learned about different functional groups. Now here's an example. Identify the functional group. We have an amino group, so NH2, that's the amino group over here. Over here we have a C double bond O, OH group, that's a carboxyl group. And well, this is an organic acid. This is an amino group, so we call this thing an amino acid. We do have another carbon in between here that can have various attachments. For sort of a variable right here. Okay, so that's the basic structure of an amino acid. Okay, what's interesting about organic chemistry is that you have these building blocks that can form very large polymers, and so we have these macromolecules that are polymers that are put together by stacking up all these monomers and bonding them with one another, so repeating monomers. And the way this works is that we can take water out of the system and combining 2 building block molecules to form one larger one. So this is called dehydration synthesis, and it's something that you definitely need to know. So in a dehydration synthesis, we're taking 2 building block compounds, let's say amino acids. We're taking H2O, a hydroxyl group and another hydrogen out of the system, which we then take out of the system as water, as H2O, and we're combining the two organic building block molecules. We're combining them to form a dimer, basically. Now, this reaction is reversible. If we're going to go the opposite direction, that means we're putting the water back into the system. You can see here, then we call that hydrolysis. Okay, so one more time. When we are taking two building block molecules and we want to put them together to make form a larger molecule, then we do dehydration synthesis. We're taking water out. Some people also call that condensation reaction, same thing. If we want to break larger molecules apart, then we do hydrolysis, and we're taking the water, putting it back into the system, and we're going to break these building block molecules back up to make them monomers. And let's move on. So carbohydrates will be our first group of organic molecules that we'll take a look at. Carbohydrates are probably the most popular source of cellular energy, so most of the carbs we burn down to make ATP, but also there are some carbs that have that serve in cellular structures. In the carbohydrate group, we have sugars and starches. They consist of these hydrocarbon chains with oxygen, and they have the summary formula of CH2ON, and that's enzymes. So basically, that's how the name came about. Carbo for the carbon here, hydrate, the water, H2O, but you will see it's not exactly water that's surrounding each carbon. But the summary formula is for every carbon, you have one oxygen and two hydrogens. Many carbohydrates, they have isomers, sort of D sugars, L sugars. You may have heard of sugar substitutes. Those are sugars that are isomers and you can't digest them, so therefore they have no caloric value. They don't add calories to your diet, but anyway, that's a sidetrack. So let's first take a look at monosaccharides, simple sugars, simple building blocks of carbohydrates. Anywhere between 3 to 7 carbons, and good example is always glucose or deoxyribose, which is used in DNA. So those are very common sugars, monosaccharides. Disaccharides, those are two monosaccharides put together, and so by dehydration synthesis, we're taking water out and we're putting two monosaccharides together, and then we get a disaccharide, two monomers put together to form a disaccharide. You can also break that back down, and that will be our hydrolysis. We actually put the water back in, and then you can break down the disaccharide back to two monosaccharides. So here is the example. We're gonna take glucose plus fructose. We're going to take H2O out, one OH and another hydrogen from this fructose molecule, and we're going to form a glycosidic bond. That's this little oxygen bridge that you see here, and that is known as sucrose. This is household sugar. We have one molecule of water. We can also go the opposite direction. We'll take the water and put it back in here and break the sucrose back up into fructose and glucose. And so the hydrolysis reaction would be to put the water back in and break the sucrose into fructose and glucose. The dehydration synthesis is taking the water out and making the sucrose from combining glucose with fructose. And so here you have the two different reactions. Dehydration synthesis goes in this direction and hydrolysis goes in that direction, just to be clear. Now we can do that dehydration synthesis many times, and if we're adding many building blocks together, then we're going to end up with polysaccharides. And so these can be tens or hundreds of monosaccharides, or even thousands of them put together by dehydration synthesis. And here are some examples of polysaccharides: starches, glycogen, dextran, cellulose. These are all polymers of glucose that have formed polysaccharides. Moving on to lipids. Lipids, phospholipids that is, are primary components of cell membranes. They consist also of these hydrocarbon chains. They also have various amounts of oxygen. And one thing that all lipids have in common, besides the fact that it's a much more diverse group of chemicals, but one thing they all have in common is that they all are nonpolar. They are hydrophobic. They hate water. They do not like to dissolve in water. They're insoluble in water. Okay, here some simple lipids or dietary fats. These are triglycerides. They contain glycerol as a backbone, and then attached will be 3 fatty acids by dehydration synthesis attached, that is. So here we're forming our dietary fat, a triglyceride. We're starting with 1 fatty acid tail first. So we're going to do our dehydration synthesis right here. We take the water out and we're adding this long hydrocarbon chain right here. So this is a fatty acid. And we're going to do this 3 times, and then we're going to have our triglyceride. And by the way, let me go back. So the other 2 fatty acid tails will be attached to this carbon and to this carbon. So then you have 3 fatty acid tails total. Okay. A little word here about saturated versus unsaturated fats. So that saturated means that we have no double bonds in the fatty acid. So if you go back to this one right here, do you see any double bonds between the carbon chain? There are none right here. So this is a saturated fatty acid, okay? Saturated with hydrogen, that is. No double bonds between carbons. Unsaturated means that you have at least 1 or more double bonds in the fatty acid tail, and that means you've got to take away from the number of hydrogens, and so therefore we're no longer saturated with hydrogen. And depending on how the arrangement of these hydrogens then is in the cis or the trans position, that's maybe a little bit too much of a technical detail, but let me show you here an example. So here we have a complete triglyceride. So all 3 fatty acid tails have been attached to this glycerol backbone. We're going to have these 2 fatty acids. They are completely saturated. This one is saturated, this one's saturated. No double bonds. Here we have a double bond. Now that takes away from the number of hydrogens I can surround the carbon with. So if I have a double bond here between these 2 carbons, I can only attach 1 more hydrogen. Now if these 2 hydrogens are on the same side, and we call that the cis configuration. If 1 hydrogen would have been on the other side, that would have been the trans configuration. Now moving on to other types of lipids here, let's take a look at phospholipids, because they're so important for our phospholipid bilayers, these plasma membranes that are surrounding cells. There we have glycerol, 2 fatty acids, and a phosphate group. And so this molecule has this polar phosphate group and a non-polar end, the fatty acid tails. And so we call this amphipathic because this molecule has a dual nature. It has this water-soluble head group that you see here in orange, and then these 2 fatty acid tails that, of course, they are hydrophobic. So you have this arrangement of 2 phospholipid rows of phospholipids facing one another. The fatty acid tails, they are attracted to one another because they're non-polar, and then you have the polar head groups that are forming the junction to the water-soluble exterior and interior to the cell. Okay, another group that's very important in the lipid category are steroids. They look like this. They have the steroid backbone, which is this arrangement of these 4 ring structures. So we have, and each corner here represents a carbon, by the way. So we have 4 6-carbon rings and 1 5-carbon ring, and then we have these various attachments, and they form an important group of messenger molecules, plus cholesterol is the sort of the mother of them all that gives usually rise to all of the other steroids. Okay, moving on to proteins. And these proteins, they are made of amino acids. And the main components of all of these amino acids, so we have lots of carbon, hydrogen, oxygen, nitrogen, and sometimes even sulfur in there. And proteins is a very diverse group of molecules. The only thing they have in common is that they are composed of amino acids, but their function is very diverse. Enzymes are proteins that speed up chemical reactions, and we have transport proteins. We have flagella proteins. Toxins are proteins a lot of times. So those proteins are a pretty amazing group of molecules and what they can do. So all proteins are composed of amino acids, and these amino acids, they have, well, as the name indicates, they have an amino group and are carboxylic acid group. So this organic acid group, therefore the name amino acid. And then in the middle carbon, you have a side chain attached to that. So here is your generic amino acid. We had a similar picture earlier. So here's your amino group in blue. The NH2, that's the amino group, and then you have the carboxyl group over here, the CO, C double bond O, and then an OH, that's an organic acid. So this is your generalized amino acid. That's what they all look like. The only variation comes in with this R group, with the side group. It could be something fairly elaborate, like this carbon structure here with the 6-ring carbon structure. This is sort of a cyclic side group. This is the amino acid tyrosine, and well, there's a variety of them. So here, before you go in shock, you don't have to memorize these, but you should know about the amino acids in general. And so let me tell you a few things that you should know about those amino acids. They all have a full name. So let's start with the simplest one, glycine. They all have a 3-letter abbreviation, GLY in this case, and they also have a 1-letter international abbreviation. In this case, in the case of glycine, it would be G. They all have an amino group. They all have a acid group, the carboxyl group right here, and they all have an attachment in the middle carbon. In the case of glycine, it's just a hydrogen. Let's move on to alanine. Alanine has a full name, alanine, 3-letter abbreviation, ALA, and a 1-letter abbreviation, A. And this one has, like all amino acids, an amino group, an acid group, and then an attachment in the middle carbon. In this case, it's a methyl group, a CH3. You see the principle. So the only variation is in this R group, whatever is attached here, and each one of these amino acids get their name based on these side chains. As I said, you don't have to memorize these formulas, these chemical formulas, but you should know that each amino acid has an amino group, an acid group. Each one has a full name, a 3-letter and a 1-letter abbreviation. And depending on the side chain, these amino acids have various properties. They're either more or less water soluble, or they may even add a charge. Here's one that has a sulfur in there that has some special implications. So the composition of amino acids and the arrangement of amino acids enables the proteins to do the job that they need to do. Okay, so here, continuation. It's a lovely little list. There's 20 of them. It may seem like, oh my God, this is such a long list. It's only 20 of them. And all these 20 amino acids, they make up all the proteins that cells use. Okay. Amino acids have another little trick to them. They come, like many carbohydrates, they come in stereoisomers. They have a right-handed and a left-handed version. And interestingly, with the carbohydrates, it's the D version that's biologically active. With the amino acids, it's the L forms that's found in nature, and it's biologically active and gets incorporated into those proteins. Okay, so here, a little sidetrack here on the stereoisomers. It really works like left-handed, right-handedness. So, in fact, the abbreviation here, L, left hand, and D comes from the Latin for right, which is dexter. So now we have the right-handed version of the amino acid and left-handed version of the same amino acid. Are they the same? No, they're not. They're not identical. They are mirror images of one another. And so just like your right hand and your left hand, they're not identical, but they're mirror images of one another. And nature seems to like that idea of creating these stereoisomers. Now, when you're connecting two amino acids to one another, we form something that's called a peptide bond. That's a term that you should know. That's the bond between two amino acids, and it's formed by dehydration synthesis, just like we saw before. So here's an example of that. Here we have two amino acids, glycine and alanine, and we're taking the OH group from the glycine and another hydrogen here from the alanine, from the amino end of the alanine. So there's the H2O that then ends up getting out here as water, and then we have a peptide bond that now holds the glycine and the alanine together. So that's a typical classic dehydration synthesis. Now, as we're adding more and more amino acids here by forming all these peptide bonds and adding amino acid after amino acid, we're going to form a polypeptide. So many amino acids together, they're forming a polypeptide. And just the amino acid sequence that is formed by all these peptide bonds, that is the primary structure of a polypeptide chain. That doesn't tell you yet what kind of function this thing can do. So we need to add structural levels to this. So the secondary structure is formed when amino acids, let's say about 20 amino acids, they get together and they form this ribbon structure. That's called an alpha helix, and that's a very important secondary structure. Sometimes we also form these beta pleated sheets right here, where these amino acid stretches of amino acids, they sort of flip over and they form these pleated sheets. Anyway, this is called the secondary structure of a protein. And so most important will be alpha helices and beta pleated sheets. Now, when we are putting these beta pleated sheets in alpha helices and the sort of more the linear parts of the protein, when we put all of this together in three-dimensional space, then we end up with the tertiary structure of the protein. So you have here an example. You have a disulfide bridge right here. You have a beta pleated sheet and the alpha helix, and then some linear regions right here. So that would be the three-dimensional arrangement in space for the tertiary structure. And if this protein ends up having several subunits that come together, then we even have a quaternary structure. So that would be the arrangement of more than one polypeptide chain in three-dimensional space. So that's only for proteins that have more than one subunit. In summary, right here, we have all levels of organization here for a protein. The primary structure is just the amino acid sequence. Secondary structures include alpha helices and the beta sheets. The tertiary structure combines the secondary structures, maybe even a disulfide bridge and any linear stretches. So that's the three-dimensional arrangement in for that polypeptide chain. And if there's more than one subunit, more than one polypeptide chain coming together, like here this light blue and the dark blue, then we even have a quaternary structure, which is the three-dimensional arrangement of these polypeptide chains together. Now, these proteins are fairly complex. They're also rather fragile and they can be denatured. That means that their three-dimensional shape is falling apart. You can picture that when you're frying an egg, it looks quite different and when it's still inside of the shell, and after frying it denatured, so it changed its nature, and that means that this three-dimensional shape sort of fell apart. Okay, so here a little bit another sidetrack here. Sometimes proteins can be attached to other organic structures, and so we can attach a protein to a carbohydrate, and then we call it a glycoprotein. We also can attach a protein to a nuclear structure. That would be a nucleo protein, or usually attached to a nucleic acid type of molecule. Or if the protein is attached to a lipid, then we call it a lipoprotein. Now, our final group of organic molecules is the nucleic acids. They consist of nucleotides, and a nucleotide consists of a 5-carbon sugar, a ribose or deoxyribose, a phosphate group, and a nitrogenous base, either purine or pyrimidine type of base. And nucleosides, they consist of a pentose sugar plus the nitrogenous base, so it's just missing the phosphate group, and we call it a nucleoside. Okay, so let's start with DNA. DNA is the abbreviation for deoxyribonucleic acid, and it contains the sugar deoxyribose and exists as a double helix. That means we have two strands of these nucleotides that are running antiparallel. The bases that are used, these nitrogenous bases in DNA, you need to know that, are adenine, thymine, cytosine, and guanine. And adenine and thymine, they hydrogen bond with each other, and cytosine and guanine also hydrogen bond with one another to form this double helix of the DNA. And that's of course, the sequence of the DNA. That is your genetics. That's your spells out all the proteins that you can make for that organism. So that's your genes and your genetic heritage. So here, the structure of DNA, we have these nucleotides. Admittedly, it's kind of small. So we have two nucleotides right here. This one is phosphate, sugar, ribose. Deoxyribose, and then this nitrogenous base. So that's one nucleotide right here. And then here, hydrogen bonding with that is another nucleotide right here. You see the phosphate, sugar, base component. And it's always that a purine base hydrogen bonds with a pyrimidine base. But in the end, you're going to get the typical double helix, and I'm sure you have seen this before. So typical DNA structure here, DNA ladder. RNA is slightly different. It contains the sugar ribose instead of deoxyribose. It is typically single-stranded, and it replaces the thymine base. We're using uracil in RNA. So in this case, then the adenine would hydrogen bond with uracil. That's typical of RNA. You find this only in RNA. The thymine is what you find in DNA. Now, luckily, the other two are the same between DNA and RNA. So cytosine and guanine is the same thing as with DNA. So here, the base uracil, very similar to thymine, but it does have a slight difference, therefore the different name. So in RNA, we find uracil. Anytime you see uracil in a sequence of some nucleotides, you know it's RNA. Only RNA has the base uracil. And here, check your understanding. How do DNA and RNA differ? Well, DNA, again, is typically double-stranded. RNA single-stranded. Typically, DNA contains thymine as a base. RNA contains uracil as a base. And then, of course, the sugar component in DNA is deoxyribose, and in RNA, it is just ribose. And finally, here we have a nucleotide-based molecule that I'm sure you've heard of, ATP, adenosine triphosphate. So adenosine tri-T-phosphate, ATP, made of ribose, adenine, and 3 phosphate groups. And let's take a look at this molecule right here. So we have typical nucleotide structure. We have the base adenine, then the sugar ribose, and then the 3 phosphates. The bond between the second and third phosphate has a lot of energy, and so that's why it's used as an energy transfer molecule. We call it sort of this. ATP is the energy currency of the cell, and it powers all these cellular mechanisms. Here, ATP, it stores the chemical energy, and it's sort of the energy currency in the cell, and it releases the phosphate group by hydrolysis. It releases a phosphate group, and that is useful energy in the cell, can be used to power other things. So here's my final slide here for this lecture, and it shows you an adenosine triphosphate, adenosine plus. So remember, adenosine means we have the adenine base plus the sugar component, so that's adenosine, and then 3 phosphates. And then by hydrolysis, we can split off the third phosphate right here, and that releases a significant amount of energy. So ATP, of course, provides more energy for the cell because this hydrolysis of the splitting of the third phosphate releases a bunch of energy. And it's a nice system because it's rechargeable, it's reversible. You can recharge your batteries basically. So you can make ATP from ADP plus adding phosphates and some energy. You can recycle everything. I apologize for this rather lengthy lecture, but this is it. You suffered through the chemistry part. You're done. So please make sure that you study this. If you're not super familiar with the chemistry, then I can provide you with some study sheets and some maybe worksheets that you can work through. But please listen to this lecture, even though it was lengthy, and kind of take your own notes on the important stuff.